So I really want to thank actually Lucy and Kristen for their introductions because they made my introduction a lot simpler. So what we know about evolution is that it is at heart a genetic process. And there are two parallel pathways of evolution. The first are single nucleotide variations. Now these are changes in one nucleotide at a time. And these change DNA and protein function bit by accumulating till eventually you do get a modified or changed function or regulation. Now the second type is what we call a gross chromosomal rearrangement. Now these are changes of large segments of DNA where they either invert, amplify, or completely disappear. Now, these can affect evolution in multiple ways, the first of which is you change copy number, which gives you a rapid change in expression level, or you can reassort gene and regulatory elements, or, and this is perhaps the most important way, is by providing redundant sequence. So if you have an essential gene that the organism needs to survive, but by amplification you get a second copy, then that organism is free to make changes in the second copy while retaining the essential function, until eventually you have the changed function taking over. So what I'd like to introduce to you today is the phenomena of environmental stress-induced mutation. So like we hear about Darwin, we hear about survival of the fittest, where you have random stochastic mutations in a genome that are selected for by the environment because they confer some advantage. But what we've actually been elucidating over the last 20 to 30 years is that the environment plays a very large part in how these mutations are formed. So you have a microbe that is specifically adapted to its environment, such as E. coli to Earth. When you place this microbe in a different environment, a different pH, a different temperature, a different carbon source, this organism becomes stressed. This stress induces stress responses. Stress responses are large collections of genes that together serve individual functions that help that organism adapt to that stress. Well, when you induce these stress responses in microbes, you find that you transiently increase mutation rates by orders of magnitude until such a time that a mutation is made that allows that organism to adapt to that stressor. Mutation rates then fall back to normal, leaving you with an adapted, evolved life form. So what we in our lab do is study the mechanisms of stress-induced mutation, aiming to understand the inherent molecular mechanism behind the formation of these mutations. And today I'm going to present to you some new discoveries where we found that reactive oxygen species is required for environmental stress-induced mutation, and I'll discuss not only how they promote mutation, but what this means for the evolution of microbes under stress. So we work in a well-characterized system that selects for starvation stress-induced mutants in E. coli. So you have an E. coli cell that has a complete deletion of its lactose genes. This means it cannot use lactose as a carbon source. It can't eat it. And then on its conjugative plasmid it has a LAC+1 mutation. So this is the LAC genes with an extra nucleotide. So these cells are phenotypically LAC-. They can't eat lactose. So when we place them in a lactose-only environment, the only cells that are going to form colonies are those that have made compensatory mutations. So what you see on right here, laser pointer. What you see right here is LAC+ colonies over the total cells plated over days of incubation. So we grow up our cells in a medium that they can use, they can eat until they reach a starvation, very dense population. At this point, we put them in the presence of only lactose and count the number of colonies or mutants that arise on days 2 through 7. Now the great thing about this system is it can detect both CNVs and GCRs. So CNVs, those single nucleotide variations, are going to be -1. They just lose a base pair. You reset, you get all of the lactose metabolism activity you need. A GCR, what the cell does is it actually takes this allele and amplifies it until you get 20 or more copies. You have just enough activity to survive. And so what we can do is we can actually detect, you take the total LAC+ population, you put them through a colorimetric assay, and you can see that the CNVs, which are very stable mutations, give you solid blue colonies, where the GCRs give you these fractioned colonies as the array breaks down. So I'm not going to get into the really hardcore genetic mechanism behind these mutation formations, but you need to know a couple things. First of all, these mutations require 3 stress responses in E. coli. Now these sense the stress. These see that E. coli is in an environment it cannot survive in. These stress responses turn on error-prone DNA translesion polymerases. So rather than the regular polymerase that replicates the genome in a very faithful manner, these are prone to inserting mutations even when they're on an undamaged template. And lastly, you need DNA transactions. You need a DNA double-stranded break, which is where both backbones of the double helix break, and then you need the DNA damage repair event to repair that break. So in a healthy, growing, easily replicating cell, when you get a DNA double-stranded break, it repairs it in a very faithful manner. But in a starving cell that has all these error-prone polymerases upregulated, you get error-prone repair of that break, you get mutations, and then that's how you get your lac plus mutants. So the big questions our lab tries to answer is 1, how does stress regulate evolution? And 2, what dictates the choice between a gross chromosomal rearrangement and a single nucleotide variation? So we have a new component of this mechanism emerging where a damaged base in the DNA is required for stress-induced mutation, and this is an unexpected constraint on both the mechanism of single nucleotide variation and GCRs. So while we were investigating the nucleotide-associated proteins, we discovered that deletion of DPS inhibits stress-induced nucleotide variations and GCRs. So what you're going to see, and this is the kind of data you're going to see the rest of the talk, is where you have rates of mutation formation normalized to wild type. So what you can see is that when we delete DPS on the right, we get more mutation. So whatever DPS is doing normally, it's inhibiting mutation. And we find that this holds true for both CNVs and GCRs. So I'm not going to split up the data for the rest of the talk, but I want you to keep in mind that everything I tell you is true for both types of mutations. So then we wanted to make sure that we weren't allowing an alternative mutagenesis pathway to happen. So we added deletions of known required genes, such as that RPOS that I talked about, and that ZenB error-prone polymerase I talked about. And when you put those in a DPS deletion background, you lose all enhanced mutagenesis. So all of that extra mutations were our characterized error-prone double-stranded break repair. So what is DPS? DPS is a nucleotide-associated protein. These are the closest E. coli has to compaction proteins. This is the closest they're ever going to get to a histone. It's induced by RPOS in the stationary phase, and it does 2 things. It physically co-crystallizes with DNA in a very unique hypercondensed way. And 2, it protects against reactive oxygen species by creating protein shells around ferrous iron to sequester them from the cytoplasm. And then, if Fenton reactions do occur, they occur within that protein shell, and DPS actually forms a tryptophan radical instead of releasing it into the cytoplasm to damage other macromolecules. So we wanted to see, how does DPS suppress mutagenesis? So there really are just 2 options. It's a physical protection or it's a chemical protection. So we went for the chemical because, frankly, the chemical was easier to test. So we looked and see whether or not the role of DPS is protection against reactive oxygen species, and which means that reactive oxygen species are in fact a required chemical species for mutagenesis to occur. So the first easy thing we did was we added known reducing agents to our assay. We added 2,2-bipyridine, which chelates ferrous iron specifically, and thiourea, which is an ROS scavenger specifically for hydroxyl radicals. And what you see is that we get a reduction of mutagenesis in a dosage-dependent manner for both chemical treatments. And more than that, if you look at the orange set of bars, that's that DPS deletion mutant I talked about. So when we add thiourea, we are completely canceling the effect of delta DPS, our first line of evidence that, yes, DPS is inhibiting via ROS protection. So the second thing is we used 2 mutants that are constitutively active for the ROS responses. Now, what constitutively active means is E. coli has 2 ways of detoxifying ROS radicals. It has what we call the SOX-RS response, which is activated by SOX-R, that activates superoxide dismutases, which take care of superoxide. And then you have the oxy-R response, which takes care of hydrogen peroxide and hydroxyl radicals. Well, constitutive active alleles mean that even in the absence of inducing ROS, we still get high levels of expression of all of those detoxifying genes. So we're decreasing the basal level of ROS in these cells. And we find that in both cases, we dramatically reduce mutation formation. And more than that, when we add the oxy-R constitutive allele with the DPS deletion, again, we completely nullify that increase in mutation. So we know that ROS is required for stress-induced mutagenesis to occur, and that DPS is acting through ROS. So the first question is whether or not the role of ROS was to induce the general stress response. Maybe it was a signaling molecule that just told the cell, you're really stressed out, you're in a really bad shape. So RSSB is a negative regulator of RPOS. RPOS, that general stress response that we need. RPOS tags it and leads it to degradation. So when we delete RSSB, we get more RPOS, more of those stress responses, and more mutation. But as you can see, even when we put that delta RSSB in the presence of increasing thiourea dosages, we can't counter it. So whatever ROS is doing, it's not for the induction of the general stress response. So that leads us to how does reactive oxygen species cause mutation formation? Well, they are incredibly damaging biochemical species. They damage every macromolecule within the cell. But I'm going to talk to you about 2 main hypotheses. One is they are required for protein oxidation, and 2, they're required for DNA oxidation. And then we break it down into how that DNA oxidation could impact, and I'll get to that later. So the first one, the easiest one, was are ROS promoting SIM through protein damage? So when you oxidize proteins, you do alter the function of those proteins, and it was a possibility that perhaps that altered function was required for mutation formation. So we used a hyperaccurate ribosomal allele that decreases the amount of oxidized proteins in cell and found that it had no effect. So ROS are not required for protein oxidation. So the second one, does ROS promote mutagenesis through DNA damage? So the first one is, can it saturate mismatch repair? So we all know that guanine pairs with cytosine normally. Well, oxidized DNA damage normally takes the form of 8-oxo-G formation, which is oxidized guanine. When you get oxidized guanine, you have a slight propensity to mispair with adenine. So we thought that maybe we were getting so many ROS mispairs that we were titrating out mismatch repair pathways that were letting those translesion polymerases make those mutations, and they weren't getting repaired. So when we added overexpression of mismatch repair, which is the P-MUTL, with thiourea treatment, we actually got an additive effect, which means that whatever ROS is doing, whatever mismatch repair is doing, they're in completely different pathways, they're doing different things. So 2, do ROS underlie the formation of that DNA double-stranded break that we mentioned was required for mutation formation? So this is an analogous assay to that LAC+1, only this is a TET+1. So we're selecting for tetracycline-resistant mutants, which is an antibiotic. So what you see, and so what we did is we took a plasmid containing CatG, which is one of those catalases that detoxifies ROS, and we overexpressed it in cells, so we're getting lots and lots of catalase and hopefully less and less ROS. And even when we put this in the presence of a DNA double-stranded break, so we're saying, okay, we'll give you a break, do you still need ROS? Well, they did. So as you can see in that last bar, that's the overexpression of CatG in the presence of a DNA double-stranded break. So again, whatever ROS is doing, it's not substituting it's not through double-strand break formation. So lastly, we said, okay, maybe it's to create a damaged base, and that damaged base, the presence of it, is required for mutations to form. So we did the same kind of experiment, only instead of overexpressing catalase, we overexpressed mut-M. Mut-M encodes for a DNA glycosylase that specifically removes 8-oxo-G from DNA. It's very specific for 8-oxo-G. So what we found is that when we overexpress mut-M and we increase the amount of excision of 8-oxo-G, we lose almost all of our mutation. So from this, we know that ROS is promoting SIM through damaging DNA after double-strand break formation, and that to be effective, those ROS damaged bases have to remain within the DNA. So from this, I hopefully have convinced you that not only are reactive oxygen species required, it's specifically required to create 8-oxo-G lesions and for those lesions to remain unrepaired in the DNA. So a possible mechanism, we know that damaged bases are replicated across by translesion polymerases, that these polymerases are error-prone, and that we require them for mutation formation. Previously, we thought they were creating mutations by acting on an undamaged template. It was just kind of a stochastic mutation formation. But now what we're thinking is that maybe, since these are starving cells, don't have replication going, we need the DNA double-stranded break to start replication again, but that these translesion polymerases aren't called into action until they come to an oxidatively damaged base. So we're going to actually test that by seeing whether or not the translesion synthesis activity of DIMB is required for mutation formation. Now, what does this mean to the astrobiology conference that I'm at? So microbes are the first forms of life on a new planet, right? The main thing that's going to drive their evolution is the environment that they're in and the stress that that puts them under. Now, we think it's pretty unlikely that reactive oxygen species are universally required because there are plenty of environments where oxygen's not present. But it is possible that an unrepaired, damaged base is universally required for environment stress-induced evolution. So we're going to test this by taking away oxidation and damaging our DNA in another way, like alkylating agents, and see if we can suppress this requirement. But altogether, this shows that a requirement for unrepaired, damaged bases, if general, would appear to present a constraint on the mechanism of evolution across all organisms in multiple types of environments, that DNA damage be present and persist. So with that, I'd like to thank both of my labs and, of course, NASA for all their money. Thank you, NASA.
Do we have some questions from the audience?
I'm a little confused as to how the GCR pathway is helpful, in particular because you've got like a single insertion mutant, right?
Yeah.
So if you just make more copies of that gene, you're just going to have more genes that are frame shifted and going to make nonsense proteins. How is that good? How do the cells survive via that mechanism?
Okay, so you get that 1% to 2%, it's a frame shift. So every so often you get a slippage, you get a read-through, you get a functional protein. So it's not like we're getting a lot of nonsense proteins they just don't make anything. So if you've got 20 copies, 30 copies, 60 copies of this thing, and each one of them kind of reads through 1% to 2% of the time, it works. And more than that, GCR is a lot of what we see in things like cancer formation. Things that lead to these increased mutation rates are actually amplification of like caretaker genes and gatekeeper genes. SMV is kind of not the predominant one for that. So.
We have a question from SaganNet.
Ooh. Yes, we do. Let me make sure you guys can hear me. One sec. Can you hear me? Okay. This comes from Donald Burke. He asks, what mutational densities can you achieve through these stresses?
I'm sorry, can you say that again?
I will say that again. What mutational densities can you achieve through these stresses?
Through these stresses. Not really sure what he means by mutational densities.
Okay, I'll ask him to clarify. I'll
Okay.
take another one. Okay, can the mutational density get high enough to create a library of mutated proteins for applied evolution of the proteins?
Man, the best I can tell you is that in our assay, we have looked to see whether or not some of these stressed and successfully mutated genomes have other types of mutations and other things that, we look for auxotrophs and things like that. I hope I don't get in trouble for mentioning this. The quick answer is we don't often find them. Usually we find the mutation that fixed it and everything goes back to normal. So,
Thanks.
I hope that helps.
Hi, fantastic talk, by the way.
Thank you.
I was just curious, you mostly talked about the conditions of the general stress response. I was just curious if whether or not this sort of phenomena, the reactive oxygen species and the higher rate of mutation would also necessarily occur in, say, a more specific stress response like the stringent response under amino acid scarcity.
Oh, that's so awesome. That's a good question. So in terms of stringent response, I don't think I'm at liberty to talk about that, but keep watching publications and maybe you'll see something. The great thing about it is that the genetic players that I talked about today, RPOS, RECA, all this stuff, we don't just see it in E. coli in this assay. We see it in bioresistance in salmonella. We see it in multi-class antibiotic drug resistance. We see it in hypoxia-induced cancer tumor initiation and progression. So the things that I'm talking about, multiple types of stresses, multiple microbes, multiple environments, this looks to be like a fairly conserved pathway for a lot of different things, which makes it even cooler, right? So.
Thank you. And with that, I would like to have one more round of applause for this morning's talks.